Assalamualaikum warahmatullahi wabarakatuh. My name is Noorisa Safia bin Abdullah. My matrix number is 227672, and in this video I will talk about what is aminoacyl-tRNA synthetase. So, what is aminoacyl-tRNA synthetase? Aminoacyl-tRNA synthetase is an enzyme that covalently couple each amino acid to its appropriate tRNA molecule. So, aminoacyl-tRNA plays a crucial role in charging process, which we will talk about in this video. So aminoacyl-tRNA synthetase catalyzes the formation of charged aminoacyl-tRNA during protein synthesis. The second role of aminoacyl-tRNA synthesis is make contact with the nucleotide in the anticodon loop in the amino acid accepting arm that are specific to the correct tRNA.
Next is structure and chemical properties of aminoacyl-tRNA synthetase. So the first one is they have binding site for the specific amino acid, matching tRNA, and ATP required for charging tRNA. The active site of the enzyme is highly specific for a particular amino acid, which ensures that only the correct amino acid is attached to its corresponding tRNA. The second is that they have two domains, which is catalytic domains and tRNA binding domains. Catalytic domains that bind the amino acid and ATP, facilitating the formation of amino acid adenylate intermediates. And the second is tRNA binding domains that recognize and bind the correct tRNA based on its unique structural feature and anticodon. So the next is chemical properties, which is the amino-tRNA synthetase is a protein that made of many amino acids. So this enzyme that have ability to recognize tRNA relies on hydrogen bonding, ion interaction, and shape complementary. So the use of ATP that provide the energy needed for the activation of the amino acid and subsequent transfer to tRNA. The high energy bond is used to link the amino acid covalently to a growing polypeptide chain that determined by base pairing between anticodon and charged tRNA. Okay, next.
Now I'll be explaining about the charging process, which involves aminoacyl-tRNA synthetase. As we all know, aminoacyl-tRNA synthetase plays a crucial role in charging process, which also involves in the process of translation, which is the formation of protein. So, for a tRNA molecule to carry out its role as an adapter, it must be charged with the correct amino acid. But how does each molecule recognize the most suitable amino acid for its partner? So this is where charging process comes in handy. Each synthetase couples a particular amino acid to its corresponding tRNAs. Then the anticodon on the charged tRNA molecule will form the base pairs with the appropriate codon on the mRNA. Overall, the charging process ensures a rapid translation of mRNA to proteins. Errors in the charging process can lead to the binding of unsuitable amino acid, which can alter the functions of proteins which form after the translation. Next, I will be explaining a little bit more in depth on the charging process through this charging animation. So firstly, a specific aminoacyl-tRNA synthetase will bind to a particular amino acid and ATP. Since amino acid has a specific binding for these two molecules, a covalent bond will form between AMP and a carboxyl group of amino acid, releasing pyrophosphate from the complex. A specific tRNA that has an anticodon to the amino acid will then bind to the enzyme complex. The bond between amino acid and AMP will then be broken, releasing AMP from the complex. At the same time, a covalent bond between amino acid and the 3' end of the tRNA will be formed, and the complex is now called charged tRNA molecule, which is ready for translation and formation of protein.